Good afternoon, everyone. All today, I Nirbhay Sharma stand before you to talk about Agrobacterium mediated gene transfer method. So, first of all, what is a gene transfer method? Gene transfer methods are basically techniques we use to introduce our genetic material into the host. Like this is your GOI. We need to introduce into a cell, maybe a plant cell, maybe animal cell. So these methods help in this transfer, making your cell competent. So first is indirect method. In indirect method we come vector based method. So this vector based method involve vectors. These can be viral vector, bacterial vector, etc. Then come direct method. Direct method like particle bombardment. In this we will use ballistic gun to insert our GOI into the cell by bombarding it against the cell. Electroporation, use of electrical impulse, microinjection, chemical mediated method, liposome mediated method, etc. But we are not going to talk about direct methods here. We will going to talk about one of the most famous indirect method, that is Agrobacterium mediated gene transformation. So for this Agrobacterium mediated gene transformation, we need to know what is a Agrobacterium tumefaciens. It is a gram-negative soil-borne bacterium which is rod-shaped, now known as rhizobian radiobacter, and causes tumor-like growth. This tumor-like growth is known as crown gall disease. Then for causing this tumor-like growth, most important is the Ti plasmid, which has phytopathogenic function. It is of around 200 kilobase pair and have four important regions: tDNA region, virulence region, opine catabolism region, and ori start sequence. So we will now talk about each in detail. Talking about the tDNA region. For example, this is your tDNA region. So what this tDNA will have? This tDNA first have 23 to 25 base pair repeats on each side, known as the left border region and the right border region. Then we have genes. Genes for auxin, cytokinin, and opine. This auxin and cytokinin result in proliferation of the plant tissue, causing tumor-like growth, as there will be increase in the auxin and cytokinin level of the plant. And the opine act as the food for Agrobacterium tumefaciens. These may be amino acid with keto acid or amino acid with sugar. Then we talk about some of the common opines such as pyruvate plus arginine, octapine, where pyruvate plus lysine give lysopine, alpha ketoglutarate plus arginine give nopaline. Then talk about the virulence region. Virulence region. See, this is a, for example, this is a virulence region. So we all know, like, our tDNA needs to be transferred. So someone needs to send the signal. This vir DNA region will send this signal to the tDNA to excise a single strand of tDNA, then making sex pilus, and ultimately the integration into the plant genome is also done by these Vir genes. Then there are 9 Vir genes, and like vir A, vir G, B1, C1, D1, D2, D4, E1, E2. Then opine catabolism, as the name suggests, it helps in the catabolism of opine in the orine region, which is the origin of replication. Then we talk about how this Agrobacterium tumefaciens causes this crown gall disease formation. So this is your plant. For example, this is your plant. What will it do? It will secrete phenols when it is wounded. Phenols such as acetocyanogen and alpha hydroxy acetocyanogen. These phenols are defense for the defense against bacteria. But Agrobacterium tumefaciens uses as signal for induction of these Vir genes. So what these Vir genes will do? These Vir genes will further stimulate excision of this tDNA, formation of sex pilus, stable transformation of this single-stranded tDNA, and ultimately integration of this tDNA into the host. So now we have talked about how this tDNA of the Ti plasmid cause tumor-like growth. Now we will talk about how we can use this Ti plasmid as a vector system. So what is a vector? So as the name suggests, it will carry our desired gene of interest. But there are certain limitations. First of all, it's large in size. Large in size, therefore low copy number. Low copy number, thereby we can't amplify it much. Then tumor inducing property, and at last absence of unique restriction site, thereby difficult to manipulate. As it is difficult to manipulate, thereby it is not advised to use. So like this, this is your Ti plasmid with auxin and cytokinin gene. We will cut this genes from here and insert our GOI into its place. It also has selection marker, plant selection marker, so that we can identify which plant have been transformed and which haven't. Then bacteria selection marker, opine catabolism region, ori site, virulence region. So to overcome this, we came with a binary vector system. Binary vector system, as the name suggests, have two plasmid. One is the binary plasmid, other is the helper plasmid. In the binary plasmid, we have a GOI, replication of Agrobacterium, replication of origin of E. coli, and there is this absence of Vir genes. You can't see any Vir genes here. And then we talk about the helper plasmid. Helper plasmid only have your virulence region and origin of replication of Agrobacterium. So like this is your binary vector. What we will do, we will insert this binary vector into E. coli. Here it will replicate and get amplified. Then we will isolate this binary vector, and then there will be another vector, Agrobacterium. It will already have helper plasmid, and we will insert our binary plasmid into it. So when there will be secretion of phenols, this helper plasmid will help in the secretion of this tDNA, which carry our GOI into the plant genome. So here we can see a schematic diagram how it is followed. We have Agrobacterium. We isolated this T8 plasmid, inserted our GOI, then transform it back into Agrobacterium, transformed into plant, selection of the plant, and then we culture those plant, and ultimately we have a plant expressing our GOI. Thank you.